Welcome back, friends. Today we will discuss about the questions on amino acids. Questions are first question: relationship between the titration curve and the acid-base properties of glycine. 100 ml of solution of 0.1 molar glycine at pH 1.72 was titrated with the 2 molar NaOH solution. The pH was monitored and the results were plotted as shown in the graph. This is the graph, and the key points in the titration are designated first to the fifth. See, key point. This is the first point, second point, third point, fourth, and fifth point. Okay. For each of the statement A to A, identify the appropriate key point in the titration and justify your answer. First point is glycine is present predominantly as the species NH3 positive CH2 and COH. The first point is point first. Okay, this species is present here. The average net charge of the glycine is positive point second. Why? Because at pH 2.34, there is an equal mixture of the NH3 CH2 COH and NH3 CH2 CO negative. The average of the CO positive, the average of the net positive charge is 0.5 or positive half. The half of the amino group are ionized. Which one? Where? Fourth point. C. Here the half of the amino group are ionized. So fourth point. The pH is equal to the pK of carboxyl group. Second point. The pH is equal to the pK of protonated amino group. This is the fourth point. And glycine has its maximum buffering capacity, point second and point third or point fourth. Why? Because these 2 points, ionization states of glycine occurred. So these 2 points has the maximum buffering capacity. Then next, the average net charge of the glycine is 0. Here, point third. In this point third, the zwitterion form of the glycine, that is NH2 positive, SCH2 and CO negative. So this point has 0 net charge. Then carboxyl group has been completely titrated. This third point. Glycine is completely titrated. Fifth point. Titration completed here. The predominant species is NH2 positive, CH2 and CO negative. That is zwitterion form. That is the third point. Then the average net charge of the glycine is -1. Last point. Why? The negative charge of glycine obtained at above pH of Pi. So last point, fifth point, has a net charge of glycine -1. Glycine is present predominantly as a 50:50 mixture of the NH3 positive CH2 COH and NH3 positive CH2 CO negative. The point second here point. This is the isoelectric point. Which one is the point third? This is the end of the titration, point fifth. There are the worst pH region for buffering power, point first, point third, and point fifth, because at these points there were no ionization state form. Ionization state of the glycine form. So these 3 are the worst pH region for buffering power. The next question, ionization state of histidine. Each ionizable group for an amino acid exchange of the 2 state, charge or neutral. The electric charge on the functional group is determined by the relationship between its pK and the pH of the solution. This relationship is described by the Henderson-Hazelbach equation. The questions are: histidine has a 3 ionizable functional group. Write the equilibrium equation for its 3 ionization and assign the proper pKa for each ionization. Then draw the structure of histidine in each ionization state. What is the net charge on the histidine molecule in each ionization state? Draw the structure of the predominant ionization state of the histidine at pH 1, 4, 8 and 12. Note that the ionization state can be approximated by treating each ionizable group independently. Then, what is the net charge of histidine at the pH 1, 4, 8 and 12? For each pH will histidine migrate towards the anode or the cathode when placed in an electric field? All three questions have one answer. For that purpose, we have to know about the structure and ionization state of histidine and their pH. So I will explain here. See, this is the structure of histidine. This structure contains three functional groups. First one is carboxyl, second one is amino, and third one is the imidazole group. And so these have the positive to net charge. Then ionization formation. Then in this second structure, one hydrogen group removed, that is protonation, protonated carry. So this structure has positive one charge. Next ionization state, in third structure, one hydrogen ion removed from the imidazole group, that is Zwitter ion form. So this structure has zero net charge. Okay. Then last ionization state, in these one hydrogen group remove, sorry, hydrogen ion removed from the amino group. So this structure has negative one net charge. Okay. This process carries in a different pH. Second structure form at the pH 1.H2, third structure form at the pH 6, and third structure form at the pH 9.70. Okay. Now the questions in question, we found a different pH. In question, we found different pH 1, 4, 8, and 12, and which structure found at a different pH. So, first structure at pH 1, second structure at pH 4, third structure at pH 8, and fourth structure at the pH 12. And net charge positive 2, first I have already told about first and second. The first structure has a positive 2, second structure has positive 1, third structure is deuterium, so 0, and fourth structure has negative 1. And migration towards those structures have positive charge migrated towards the cathode, and those no have charge, so migrate towards the there is no migration. Then structure has the structure has a negative charge, migrate towards the anode. This all knowledge regarding histidine, their ionization state, and pH. This all about the answer of this question. Okay. The next, separation of amino acid. Next question is the separation of amino acid by ion exchange chromatography. Mixtures of amino acid can be analyzed by using the first separating the mixture into its component through the ion exchange chromatography. Amino acid placed on the cation exchange resigns containing the sulfonate and sulfonate group and flow down the column at the different rates because of the two factors that influence their movement. Two factors are ionic attraction between the sulfonate residue on the column and positively charged functional group on the amino acid, and aggregation of the non-polar amino acid side chains with the hydrophobic backbone of the polystyrene resin. For each pair of amino acid listed, determine which will be the eluted first from the cation exchange column by pH of 7.0 buffer. See, here resin is a cation exchange resin containing sulfonate group. Okay, and also given the two factors. Now question is, from this pair of amino acid, which one elute first? So for that the question, we have to know about the principle of ion exchange chromatography. So the principle is very simple one. Proteins move through the column by their net charge at a given pH. Okay. Then with the cation exchanger, protein with a more negative charge move faster and elute first. Protein with a negative charge, high negative charge or more negative charge move faster and elute first. This is the principle. Now from this pair of amino acid, first one is the aspartate and lysine. Aspartate is a polar uncharged R group and lysine is a non-polar aliphatic group. So aspartate elute first. Then arginine and methionine. Arginine is a positively charged R group and methionine non-polar R group. So methionine is a elute first. Then glutamate and valine. Glutamate is a negatively charged R group and valine is a non-polar aliphatic R group. So glutamate elute first. Glycine and histidine. Glycine is a non-polar aliphatic and histidine is a positively charged. So glycine elute first. Serine and alanine. Serine is a polar uncharged R group and alanine is a non-polar aliphatic R group. So serine elute first. This is about ion exchange chromatography. Then naming the stereoisomers of isoleucine. The structure of amino acid isoleucine. Given here. This is the structure of amino acid isoleucine. How many chiral center does it have? These have the two chiral center. This one is the first and this one is the second. Alpha carbon atom is the chiral center. Okay. How many optical isomers? This isoleucine have the 4 optical isomers. L-isoleucine, D-isoleucine, L-allo-isoleucine and D-allo-isoleucine. Okay. And draw the perspective formula for the optical isomers of isoleucine. Here, these are the structures of optical isomers of isoleucine. Same for threonine and 4-hydroxyproline have the 2 asymmetric C atoms and thus it has 4 isomers. Thank you.